Good morning, everybody. I'm Kent Simmons. I'm a conference director with Cambridge Health Tech Institute, and I would like to welcome you all to day 3 of the second annual PEG Summer Symposium. We're very happy to have you with us today. Just looking at those in attendance already, I see at least 5 time zones represented, so it's very exciting to be able to share this meeting with those of you from around the world today. It's morning here on the East Coast of the U.S., but for some of you it is later in the day, and some of you much earlier. So we hope you have some nice coffee going wherever you are. Today we'll be encouraging you to ask questions in the public chat, and you'll have kind of 3 different types of opportunities to do that. During each of the recorded presentations as they play, you'll see you'll see that the speakers are online during that time, and you'll be able to ask questions there. This will be in the chat box to the right of your screen. At the ends of their talks, the speakers will come on live. You'll have 5 minutes or so to ask questions there, again in the chat. And then at the end of each of these sessions, at noon, and then again at the end of the day in the afternoon, you'll be able to come on, and all of the speakers from that segment will be involved in a panel discussion, and you'll be able to ask questions there also. For your future reference, the presentations we have approval to share will be available on demand to registered attendees 24 hours after the live sessions have concluded. And we'll send you an email with a link, probably the same link you have here now, to allow you access to those. You should, if you're registered for the full week, you should have received those links already for the day 1 and 2 sessions. Day 1 was on engineering themes, and yesterday was on oncology. Tomorrow, which will begin at 9 o'clock, will be on themes related to expression, and all of those current agendas are on the website for the meeting. Those are the most up-to-date agendas, so please have a look there. Nobody is canceled. We've made some fine-tuning adjustments here and there to the time schedule. Couple of reminders before we get started today. All of us here at CHI are very excited to be returning again to in-person conferences starting next month in Boston with our Bioprocessing Summit meeting. But I know a lot of you are fans of the PEG series of events, and I wanted to tell you about our upcoming in-person conferences in the PEGS world. PEGS Europe this year will be back in Barcelona on November 2nd through the 4th. I think you'll be able to see those programs on the website now. And then PEGS Boston will be May 2nd through the 4th at the Heinz Convention Center in Boston. We're just starting the research process for that now, and those programs and preliminary topic outlines will start to show up on that website soon. So I'd like to introduce our first presentation for today. This is Harsha Gudamwardena. He's a senior scientist in the mass spec group of Janssen. He'll have a talk that will go for 20 minutes or so, and then be available at the end live for your questions in the chat. And Harsha is also online now to answer any questions during the playing of his presentation. So I'll be back with you after the presentation, and thank you again for joining us today.
Welcome, everyone. First, I would like to give my sincere appreciation to the organizers of this symposium for giving me this opportunity to present our work. In today's presentation, I show how emerging tools and technologies can be leveraged to solve problems in protein characterization. I will focus on how technological innovation can provide both comprehensive and rapid characterization of biologics. Here's an outline of my talk. First, I will give a brief introduction to my group's role in large molecule analytical in early development. Next, I will describe several different tools and technologies used in biologics characterization in a stage-appropriate manner throughout the development life cycle of biologics. Finally, I will conclude my talks by summarizing our findings and what we hope to accomplish in the future. The figure represents the general stages in large molecule early development. Once a target is declared during pre-portfolio research, candidates in the range of 100 to 1,000 molecules get screened and filters through lead generation and lead selection to less than a dozen molecules per program that enters our department, the Cell and Developability Sciences, or CDS for short. The CDS group is responsible for the selection of the cell line that goes into late-stage development and is a function that sits in between discovery and development, as seen in the gradation here of the colors. The analytical function within CDS uses mass spectrometry to characterize biological molecules generated during cell line selection activities. These molecules include, as you can see on the top left-hand corner, the typical MABs and various types of non-MAB molecules, such as drug conjugates, vaccines, as well as these multi-specific formats that come also as a 2+1 design, which are generally used in immuno-oncology. The cell line development starts with the transfection of the expression vector with the genes of interest into a host cell line. Single cells are next plated by isolating clones from pools using a process known as VIPS, or Veriside in situ plate seeding. These single clones are then expanded to 96 deep well screenings, fed batch shaped flask screenings, and final amber bioreactor screenings to finally select the top clones. The entire process takes 4.5 months from transfection to selecting the manufacturing cell. We use fit-for-purpose analytical methods shown in the table to your right that includes mass spectrometry and also other types of biophysical methods. For example, we perform in-depth characterization at the amber bioreactor stage, while high-throughput screening techniques are more amenable and used for triaging clones at the 96 deeper stage, as shown in the yellow funnel. This workflow allows some more molecules to be developed faster, where backup molecules as well as backup clones can be used to mitigate development risk. In order to meet these new deliverables, analytical functions need to innovate in new tools and technologies that increase both the speed and quality in which we do analysis. We believe that automated solutions and novel methodologies will reduce the bottlenecks in the process where CLD or cell line development campaigns can move faster to IND. This brings me to the main topic of this talk: what are the emerging tools and technologies, and how do we use them to accelerate biologics development? Here's a preview of what you will see as technologies that have helped us in early development. This is not by no means the entire list of things we do, but for this talk, I've decided to show some of these key technological innovations that we've developed in our laboratory. I will cover these topics in this order. First, I will introduce our data analysis pipeline. Our data analysis workflow has been a key to a success of our lab's ability to deliver to the pipeline. We have an end-to-end automated sample preparation and data analysis pipeline that we have implemented in collaboration with Protein Metrics and other companies. Automation of sample prep is ongoing work, and today I'll be presenting most of the data analysis pipeline using BIOS software, which is an integral part of our pipeline for the past 4 years. Key features of the software include data generated both in-house using our mass spec instrumentation, as well as from CROs, automatically subject to a vendor-agnostic bionic search, which is the search algorithm that groups peptides from their mass spec data, and extensive product variant searches of known modifications as well as unknown or unexpected modifications are performed using what's known as a wildcard search. And both these modifications are then reported simultaneously in a search. Validation of search results and automatic exports of the results are performed. And then finally, data aggregation with molecule information is performed using gene data biologics. Recently, we have integrated intact MS analysis workflows with our peptide mapping to report these results jointly and compare modifications in both the intact data as well as the peptide map data. This gives us the ability to validate results from orthogonal methods. Here's an example where BIOS is used for sequence variant analysis, or SVAs. Our peptide pipeline data sets are automatically subjected to a database searches looking for amino acid substitutions while you're looking for other types of modifications. SVAs are annotated based on the filter logic shown in the decision tree at the bottom left-hand corner. And I'm showing a rare instance where we found this sequence variant, which was a phenylalanine to tyrosine, where a spectrum matched multiple XICs. And these were tyrosines that exist in different positional isomers, as different positional isomers. So we detected the meta-ortho isomers in addition to the naturally occurring para-tyrosine isomer. Here's an example of this approach where we compare modifications of an intact antibody after mass deconvolution with a reconstructed spectrum of modification estimated from the peptide map of the antibody. The profiles show good similarity for most of the major modifications, which are the glycosylations. Intact MS data can also reveal unexpected modifications. In this case, the spectrum on the right-hand side shows a plus 340 Dalton peak, which was confirmed later as a hydroxylysine galactosyl glucose by peptide mapping. The MS2-HCD spectrum on the right shows extensive neutral losses of the sugar units of this modification, despite we were able to localize the modification onto the correct lysine residue. However, this may not always be the case. In the last example of our pipeline, we are using instruments that are equipped with electron-based dissociations to preserve modifications that are weak, such as the glycosylation. ETD is an indispensable method to localize glycosylation, while HCD, or high-energy collisional dissociation, provides information on the glycan structure. ETD is also capable of facile dissociation of disulfide bonds over the backbone bonds of a peptide, which we routinely use for disulfide mapping. Recently, emerging electron-based dissociation methods such as ECD in an EXD cell and electron-activated dissociation have also become commercially available. In the top right-hand spectra, I'm showing the electron-activated dissociation spectra that distinguishes leucine from an isoleucine isomer. And also isoaspartate can be distinguished from aspartic acid using specific reporter ions due to electron-based fragmentation. We also have interest in improving sample preparation, specifically sample digestion speeds. Microdroplet-assisted digestion was a Janssen collaboration with a group at the New Jersey Institute of Technology and at Stanford, who have actually pioneered these methods of digestion using microdroplets. Conventional digestion methods are rather time-consuming and not amenable to high-throughput characterization of biotherapeutics. Here we describe a method of generating microdroplets using a mixing tea where reactants were mixed prior to electrospray ionization, or in this specific case, which is called electrosonic spray ionization, and introduced separately allows the reactants to form after ESI. These methods are amenable to comprehensive digestion and reduction of disulfides of antibodies at unprecedented speeds of 4 seconds on the top case, where they are mixed, to as fast as 250 microseconds when they are infused separately in 2 different sprays. We have used both IDES, which is a protease that's used for subunit analysis, and PNGSF, which is used for deglycosylating glycans on antibodies, two of the most widely used enzymes to prove as proof of principle experiments, along with T-cell chemical reduction which we can do in multiple steps or as sequential reactions or in a single reaction step, what we call one-pot reaction using these microdroplets. We recently reported these results using antibody microdroplet reactions. And here I'm showing you some of the results from this paper where monoclonal antibody, Fab, and single-chain FC fragments are generated using the ICE enzyme. And we were then able to compare the same reaction that was performed in solution, which we call the bulk reaction. The top spectrum shows the completed digestion of the antibody to the Fab and single-chain FC fragments at room pressure in 4 seconds. The bottom spectrum shows the intact antibody that remains undigested after 5 minutes of in-solution digestion at 37 degrees, which is the most optimum temperature for ICE. Considering typically in-solution digestion of a mAb with ICE takes about 30 minutes and sometimes up to 60 minutes to complete, microdroplet digestions offers 450-fold increase or improvement in speed. We have also combined PNGSF deglycosylation with ICE digestion and reduction to produce subunits in sequential steps. Here, the FC fragment is free of the glycosylation, and this in particular is useful in looking at other types of modifications that are similar in mass to a glycosylation, such as glycan, and we've actually demonstrated that in this paper. We next generate antibody subunits by reduction and ICE digestion in the single pot reaction. The reaction is again performed in the T-junction, and the reaction products are collected. And the time that this entire reaction is completed is about 4 seconds. The deconvoluted mass spectrum shows the light chain, the FD fragment, which is the heavy chain fragment above the cleavage site of ICE, and the single chain FC, which is the bottom half of the heavy chain at the cleavage site of the ICE. Each one of these fragments is roughly 25 kilodaltons, and we can see all three. And the single chain FC fragment also shows the characteristic glycans. The same reaction can be also performed in real time using 2 emitters, where the second emitter needle is nebulized in charged solvent ions that reacts with neutral antibodies to generate charged fragment ions. All 3 antibody fragments are observed in 250 microseconds, which is approximately 7 million x increase in digestion speeds. One drawback to both these methods that we publish is that antibodies and enzymes are introduced using a syringe. This is rather cumbersome and not truly amenable for high throughput operations in a true pharmaceutical settings. To circumvent these issues, we have automated sample introduction by using flow injection MS analysis. This was done using an Agilent autosampler that was programmed to draw both the antibody and ICE enzymes from separate wells. You can do them where you can sandwich the sample in between the enzymes, or you can pick them separately. And then once you pick them up, you can mix them in a loop very rapidly and directly inject into the flow plot of the MS. In this setup, the isocratic flow is made up of ammonium acetate, which is also the digestion buffer and is compatible with MS. It is important to know that we have modified the Agilent jet stream source for sonic spray ionization and tuned the Agilent QTOF for high MOZ in order to observe native spectra of the antibody and digested fragments. Here's the data we get from this experiment, where the spectra were collected for 10 seconds and cycle times between samples by the autosampler is about 1 minute. In these experiments, products from complete digestion was observed. Under suboptimal conditions, we can also obtain partial digestion of a single chain, a single heavy chain, as seen in this spectra. And by tuning different parameter settings, we can actually change the product distributions quite well. We are also interested in improving sample separation speeds to accelerate biologics characterization. And I will show a couple of case studies on how we use separations technologies to speed up characterization. In the first case study, we are investigating clipping on an FC fusion protein. Clipping or fragmentation of the therapeutic can take place via non-enzymatic mechanisms as well as due to the activity of enzymes present in cell culture harvest. Identification of clip sites are important in the development of safe and efficacious therapeutics. However, looking at the clip site or localizing the clip site is somewhat challenging even with methods such as peptide mapping. Here's our workflow for clip site detection. We label the protein with a fixed charge derivative called TMPP, which is a chemical tag, a bulky chemical tag that preferentially reacts with mostly the N-termini of the protein. So this is an ideal tag to react N-termini that are present due to clipping. Then we digest the protein using trypsin, and the digested peptides that have the TMPP tag are confirmed by the detection of specific reporter ions, and we report the specific reporter that was generated exclusively with electron-based dissociation methods such as ETD. Further, in this method, reporter ions are used to trigger additional MS2 scans to confirm the sequence. And in this case, you can see that same precursor is further triggered again by HCD or CID MS2 scan. Using this approach, we examine the diagnostic utility of this reporter ion, and with over 1,000 TMPP labeled peptides from a K562 total cell digest. In addition to the highly selective nature of these reporter ions, we noted that most labeling occurs at the N-terminus and that labeled peptides are significantly hydrophobic compared to their unlabeled counterparts. And you can see in this chromatogram, and they elute during the rapid sawtooth ramps of the LC gradient, where the organic, the amount of organ is ramped. Using this information, we have completely automated the process for looking at clip sites, where assay map Bravo is used for both labeling and digestion. This workflow allows a rapid LC separations followed by targeted MS to detect the clip sites all in a time of 12 minutes. Rapid and highly selective separations are achieved through this method for detecting clipping in the FC fusion proteins. We detected multiple clip sites, which are all sequential, using this method of triggering diagnostic ion-triggered MS2 scans, as well as we can apply this to the NIST map, as well as the K562 labeled peptides, where retention time of the labeled peptide is used as also a predictor to identify these clip sites. In the second case study, we are using ion mobility separations to perform using a technique known as structures for lossless ion manipulation, or SLIM, to speed up biologics characterization. SLIM experiments were performed in a Janssen collaboration of Mobile Ion Systems, who is the exclusive licensee of the SLIM technology that was developed in Richard Smith's lab at the PNLL for commercialization purposes. In the workflow shown here, we investigate released glycans from human serum IgG loop of lupus and healthy subjects. IgG from serum is purified, and glycans from purified IgG are released and labeled. These released glycan samples are then introduced using an Agilent LC by flow injection to a SLIM device that is coupled to Agilent 6545XT mass spectrometer. I'd like to show an animation of how the SLIM operates. We introduce a 30-second long slug of sample by flow injection first. The SLIM ion path is about 13 meters long. The molecules are ionized by electrospray into the heated capillary and focused using an ion funnel. The serpentine path is a traveling wave that allows ions to be separated, and it can also accumulate ions on board and release so that ions are separated based on their collision cross sections. So what we're measuring is the arrival time of these ions. More compact ions arrive fast to the detector, the TOF detector, and the more extended ions arrive later. What's unique about this is the same mass to charge having two different conformers can be separated. And this gives us the ability to identify things that we can't necessarily detect only by mass spectrometry. The two-dimensional plots of mobility and mass to charge are obtained. And in this particular experiment, 55 ion packets are collected per 2 minutes of flow injection runs. This gives us roughly about 14 ion packets of data for the 30 seconds of flow injection. This is a comparison of HILIC-MS with slim IMS-MS analysis of released glycans. We can see that a typical HILIC run takes 15 minutes to separate glycans, whereas IMS can do that in less than a second, but with accumulation of 2 minutes of the flow injection. The table shows that many more glycan species are detected at 2 minutes of flow injection run in comparison to the 15 minutes of HILIC. So it definitely allows us to take more with less time. It is known that galactose levels are implicated in autoimmune disorders, and one of our goals was to see differences in the different glycans between lupus and healthy subjects. The low levels of galactose have been reported specifically for rheumatoid arthritis by us and others in previous studies. However, glycans derived from these autoantibodies in our analysis from SLIM does not show any statistically significant lowering of galactose. When we examine the intensity ratios between G1F and G0F, that is a measure of a single galactose, or intensity ratios between G2F and G0F, which is a measure of two galactose units. We next looked at additional isomers observed by SLIM separations. For example, G1F glycan is resolved in 3 peaks, reproducibly in triplicate measurements for all samples analyzed. We used the intensities of the 3 G1F isomeric species as a separate predictor to categorize lupus and healthy subjects. To our surprise, when we did this ROC analysis, the ROC curves of the third G1F glycan species is more predictive of the disease state compared to the first and second G1F isoforms. And there is a lot of interesting data in these differences in these glycoforms that we have identified, and we are planning to show that in a future report, in a publication. In the third case study, we are using SLIM separations to speed up characterization of antibody-drug conjugates, or ADCs. An important parameter in evaluating the conjugation of a small molecule drug to the antibody is drug-to-antibody ratio, or DAR value. Traditionally, we have obtained DAR values using time-consuming 2D LC-MS analysis. Here is an example where we are using size exclusion chromatography as the first dimension, followed by reverse trace separation in the second dimension to obtain DAR values using this multiple hard cutting 2D LC-MS technique using the Agilent 2D LC-MS system. We observed that the SEC retention times kind of correlate quite well with the DAR values, and we can just simply use the SEC to predict DAR values. We next use SLIM ADC experiment in a collaboration with the Pacific Northwest National Labs to examine a model antibody-drug conjugate. In these experiments, we were able to rapidly and simultaneously identify conjugated species and estimate the DAR values accurately of both the heavy and light chain of model antibody-drug conjugates. We were able to estimate the DAR values not only for just the heavy chain, but for each glycoform, and different drug loads for each of the glycoform were able to be accurately determined with the high resolution capabilities of SLIM. Then we combine the light chain and the heavy chain glycoform DAR values to obtain a sum average for the DAR value, and that DAR value corresponds quite closely with what's known in the literature as well as what we obtained by SEC. Multiplexing. Multiplex sample analysis is another important method that we have developed to increase throughput in our peptide map analysis. Isobaric labeling reagents such as tandem mass tags or TMT tags are reagents that are widely used in proteomics for relative and absolute quantitation. Their use in pharmaceutical characterization is rather limited. Here, we are using tandem mass tags to create isobaric label peptides to estimate the occupancy of small molecule drug conjugated to antibodies. As shown in the diagram, the workflow starts with labeling the unconjugated cysteine site of a protein of the ADC sample. These are the antibodies that have the drug conjugated. And the same antibody without the drug that we call a mock sample is also reacted with another cysteine reactive TMT reagent. The first pair of the cysteine, first reagent is a cysteine reactive TMT reagent or iodo-TMTs. These parallel samples are then trypsinized, and the resulting peptides are then labeled with a second TMT tag that are reactive towards the N-terminus and the lysines. So if of all peptides after they're digested have an N-terminus, some peptides do have lysines at the C-terminus, some don't, but every peptide we are sure to have an N-terminus will get labeled with this second TMT tag. The peptides are then mixed in equimolar ratios and analyzed by LC-MS. The peptides were subjected to MS2 generate TMT reporters from both the first and the second pairs. This is rather atypical for a standard TMT type of proteomics experiment where you're using both labels. We are using both these TMT tags and the intensity ratios between mock and actual samples to derive occupancy estimation by the equation shown at the bottom. Here are a few examples of site-specific occupancies estimated for cysteine conjugates at different sites have different occupancies as seen in the top panel. The spectra shows the reporters, and the bottom panel shows the corresponding fragment ions that localize the conjugation sites from the backbone bonds. Once we obtain occupancies of each site, we can make use of the non-cysteine peptide TMT ratios, or the spectra which have exclusively the second TMT label to obtain a normalization factor between the conjugate and the mock. This is kind of useful to normalize the entire data set to obtain accurate occupancies for every conjugate, every site-specific conjugation of the drug. So with using these normalization factors to correct occupancy estimate for each site-specific cysteine conjugation site, we are able to map them and show that each site has different amounts of the drug. Using this approach, we can multiplex up to 4 different conjugations to estimate occupancy of a drug in a single experiment. And this gives us unprecedented speeds and throughput in assessing different types of reactions, conjugation reactions, and evaluating the best type of chemistry to use for conjugation. The example shown here is a site-specific occupancy estimation using TMT 10-plex reagents to monitor up to 4 different conjugation reactions in a single run. This is kind of unprecedented given the complexity of the experiment. And here we are using the lysine or N-terminus labeling TMT reagent to do the normalization. This is an example where the normalization was very critical. You can see that you can use the lysine TMT reagents to obtain ratios that can be used to normalize the conjugation sites using this dual TMT approach. And using this series of equations, we can actually obtain accurate or corrected occupancies by normalizing with the normalized data sets. Our final approach is to speed up biologics characterization by rapid sample introduction, and there is no separation in this case. This type of analysis is particularly important for multi-specific antibodies that can produce desired heterodimers as well as the undesired homodimers during single-set cloning. In this scheme, we are showing how cell line development activities require rapid product quality analysis, specifically looking at chain pairing of multi-specific. One needs to screen large number of clones during cell line development stages to identify and rank order cell lines with the highest percentage of the heterodimers. We are using the RapidFire MS, in this case with cycle times of 8 to 10 seconds per sample to speed up the processing of large number of samples. Our approach starts with purifying cell culture material and subjecting samples mostly in 96-well format for RapidFire MS analysis. The data generated from these experiments can be split to individual files and processed using our intact MS data analysis pipeline, where the intact mass spectra are deconvoluted and intensities are reported for each clone for various species, mostly glycan species, or any type of chain mispaired species. And you can see that in the bottom heat map. Here's an example where only 13% for the high titer clones moved into cell line selection due to high levels of homodimers and half-mers, which are impurities. If a screening method was not used for triaging, we could have ended up having major product quality issues in some of the high titer clones. In addition to chain mispairing, we are also exploring the use of RapidFire MS for examining product quality attributes such as glycosylation, which is also a critical attribute, especially in the case of multispecific, that we've seen high levels of mannose-5. Here is an example of what a 96-well plate intact mass analysis looks like. In the chromatogram, we see 96 peaks collected in simply 20 minutes. Each peak contains an MS profile of the sample, and each peak in the tick is split to individual files and automatically subject to mass deconvolution using protein matrix bias pipeline. In this study, we have 10 plates run continuously for 3.5 hours. In this example, we investigated the glycans of 960 sample spots with high reproducibility, less than 1% CV, with data quality comparable to LC-MS. And the failure rate was approximately 0.4%, and all this happened in about 3.5 hours. This screen represents a general comparison in speeds. As you may have noted that RF-MS shows significant improvements in speed and throughput. However, if we are to take advantage of this high throughput analysis, the back-end data analysis also has to be synchronized with data generation. It is analogous to what's shown in this cartoon to two sprinters handing off the relay baton perfectly. This is our ultimate goal, to make this process completely automated, both from the sample generation standpoint as well as the analysis, to make it 10 times faster for faster decision making. And if this can be done for 96 well plates, you can imagine how it would scale in throughput when we do more samples, more plates. The speed improvement would be exponential. In conclusion, I hope you have seen a variety of methods we have developed to accelerate biologics characterization in a stage-appropriate manner. These enabling technologies and tools and technologies include increasing digestion speeds a several million-fold using microdroplets. Microdroplets accelerates otherwise slow reactions and is a transformative technology in the characterization of biologics with increased throughput and versatility. I've also shown short gradient LC as well as ion mobility separations to speed up analysis times at unprecedented speeds. Sample multiplexing was also discussed in the context of antibody-drug conjugates and fast peptide quantitation, and rapid sample introduction from screening to product quality assessment of antibodies was also presented. I've also demonstrated how we use alternative fragmentation methods, such as electron-based methods, to confirm sequences as well as to localize modifications. And finally, I was able to show how chemical labeling is used for detecting and localizing clip size with triggered MS2 scans. As future directions, we are continuously innovating and improving our analytical methods, informatics capabilities to become an industry leader in biologics characterization. We also see opportunities for improvement in sample management and data tracking systems, integrating high throughput mass spectrometry with robotic sample preparation, and increasing the information content of analysis by integrating various assays in autonomous fashion. Maybe we can use robotics to perform various types of biophysical assays along with mass spec assays, and that's what we're working on, and that's probably going to be our future in the next few years. We envision that these new technological innovations will provide time to value in our biologics characterization. With that, I would like to thank all the contributors at Janssen, my colleagues, especially in the mass spectrometry lab, and our research partners and collaborators. Finally, I would like to thank you for your participation and attention to my seminar, as well as I would be happy to answer any questions. And thank you.
Questions for the next 4 or 5 minutes before we begin our keynote presentation. Harsha, are you with us? There you are. Good morning.
Good morning.
It's good to see you. I'll look forward to actually seeing you in person soon, I hope.
Yeah.
So we have time for a few questions today, and to start things off, we have a question from Roderick Chalk. Roderick's our keynoter who will give the next talk, and he wondered if you can comment on the resolving power of SLIM-MS.
Yeah, so SLIM can be, it's a very high resolution arm mobility system. Dick's group has actually shown resolutions up to 700 in multi-pass modes, where you can actually use not just the commercial SLIM where you can just do 13 meters, but you can pass it many times, multiple times, and achieve that kind of resolution. But in the current commercial version that we have, we can achieve closer to 100 resolution and mobility resolution with the technology that's going to be made available to the masses. So that's where we are at the moment.
That's great. Thank you. I have another question. Are there additional reagents for derivatization of N-termini, and have these been used for investigating clipping of biotherapeutics?
Yeah, I mean, the N-terminal chemistry is quite, there's lots of reagents that can actually derivatize N-termini, which is a primary amine, and, those reagents can also label the lysine residues, which also is a primary amine. So, but in the context of clipping, it's very advantageous to use reagents that only selectively label the N-terminus. So the TMPP, the chemical structure is a bulky chemical structure, right? And that can somewhat selectively label the N-termini in addition to sometimes a few lysines and also tyrosine residues. But a majority of the peptides that we've detected in our studies show that it has very high selectivity towards the N-termini. And so that's where, we make use of this specifically to look at clipping using this N-terminal reagent, tagging reagent.
Okay, and on the same theme, are there reagents for modifying the C-terminus of a clip site?
Yeah, that's a pretty good question, because actually there are no commercially available reagents. Tagging the C-terminus is rather tricky. Mm-hmm. And there's no real way to do that selectively. You can label the carboxylic groups, but then it will also label glutamine and aspartic acids, which are also the same carboxylate groups, right? So, but there's been a recent development in the lab of David McMillan at Princeton, who has developed some photo redox chemistry to label C-termini very selectively, highly selectively. And they've demonstrated this technique on model proteins. And this has a huge potential in actually being applied to problems in biotherapeutics, where you can actually also derivatize C-termini quite selective and make use of this novel labeling methods to actually look at clipping from the C terminus. So we need a combination of both, and currently we only have ways to do that from the N terminus. Got it. Thank you. I think to keep us on schedule, we're going to move to our next presentation now. But Harsha, thank you for your talk today. Harsha will be facilitating our panel starting at noon. This will be a live panel with all of the speakers from the morning session, including Harsha. And if you have other questions for him, you could ask them there or also in the chat, and perhaps he can answer you there. So thank you, Harsha, and I'll look forward to seeing you back shortly. Thank you, Ken.